Welcome to Microbiology Department. The Department of Microbiology was established in the year 1997 with an intake of 30 students and single combination. Now the department has 160 students with 12 combinations. And here is the main vision and mission. The main vision is to produce nationally and globally competent students by sustaining their request to explore advanced areas of microbiology, excel in new biology research and contribute for the betterment of mankind. And the main vision of the microbiology is to educate and train students in the discipline of microbiology. Here is the student strength from the year 2018 to 2024. Department has well-equipped laboratories for training undergraduates. And here are the some of the department has held 27 student projects in the past 6 years. And here are the some pictures on the board. The department has conducted different programs, for example, like seminars, inter-college competitions, extension teachers, staff members, and many other activities are held in the past. The department had held different programs such as student interactions, student seminars, poster presentations, and student progression, extension lecturers, bridge course, like this many other courses were held in the department from the last 6 years. We have some of the pictures of the activities. You can show them. Some of the outstanding students who got gold medal in microbiology in the name of C. Jaya Prakash Reddy, which was established in 2005.
Apart from routine academic activities, the Department of Microbiology at all City College is also active in performing research and publications. We have presented research papers, 6, and published 8 research papers in reputed journals, and published 11 textbooks for microbiology students in the past 6 years.
Pooja, BSc Life Science, second year, autoclave. Autoclave is invented by the Charles Chamberlain in 1879. And the autoclave is a shield device which is similar to pressure cooker that kills the all microorganisms using the saturated steam under the pressure. The use of moist heat kill the all microorganisms including the high resistant endospores. The boiling point of the water varies depending upon the surroundings environmental pressure. The usual procedure to heat at 1.1 kilograms centimeter steam pressure, which is the temperatures of 121 degrees Celsius. At 121 degrees Celsius, the autoclaving to achieve sterilization can be considered 15 to 20 minutes, which depends upon the volume of the load.
So here is the equipment called centrifuge. So it is invented by Antonin Prandtl. So basically what it does is, it separates the particles from a liquid sample. So it uses sedimentation principle, which is nothing but when heavier particles in a sample, it settles at the bottom of the tube, and lighter molecules or particles rise to the top. So sedimentation is dependent on gravitational pull, and when it comes to the centrifuge, the additional force is added, that is centrifugal force, which is enhanced a bit to separate the particles. This time.
Good afternoon, myself Vijayalakshmi from GCC Life Science second year. It's a hot air balloon invented by Brice Pasteur in 1800s. It works on the principle of forced circulation, radiation, and conduction. It is used to sterilize glasswares like petri dish, tubes, pipettes, etc. We can keep the glasswares like this and flow. gas sterilization for 160 degrees is 1 hour and for 180 degrees Celsius is 30 minutes. Thank you.
Explaining the incubator. Incubator is a laboratory instrument or a heated insulated box which is used to maintain the growth of microorganisms and other cells and biological entities. The principle of incubator operates under the thermoelectricity principle. The incubator also maintains temperature, humidity, sometimes gas control. The procedure of incubator: first we open and clean the incubator and calibrate it. Then we have to adjust the temperature, pressure, and other regulators and main switch to on the incubator. And then we have to prepare and culture media and inoculate it and by using sterile techniques. Then we have to place the cultural media inside the incubator by ensuring some space. And next one, we have to check the temperature, humidity and the growth of culture. It lastly, after incubation, remove carefully the culture media and analyze it with using sterile techniques.
These are digital colony counter and analog colony counter. These are used in microbiology lab to count the number of colonies which are formed in a petri dish. To use a digital colony counter, we just have to place the petri dish on this place and then use this pen to count the number of colonies on it. When we are using this pen, the digital colony counter automatically increments the counter. This way we don't need to keep count of it and it becomes easier. This is used to estimate the number of microorganisms in a given area. The number of microorganisms is calculated in CFU by ml. Thank you.
This is an equipment used in worldwidely in microbiology, biochemistry and many laboratories. So basically what it does is, we can see some microorganisms and which can be seen by our naked eye, can be seen by this microscope. So what we do is, we place the sample in this stage and when the light is travel from this source to the condenser, the condenser condenses the light to the sample stage. Then the light from the sample stage reaches the objective lens. Then from there the light travels to the iris. There we see the sample through the iris. So basically we can see some microorganisms through this microscope. This is objective lens, which is magnification is 100x, 400x and 1000x. So we have to adjust the objective lens based on our needs. And here is
Laminar airflow cabinet. In this, we use it to keep a sterile work environment inside this cabinet. So how this works is, there are HEPA filters, high efficiency particulate air filters, which filters out all the microbes which are up to 0.3 microns, and then the air filtered out air comes out from here and falls down to this place. Here we can keep our petri plates and culture medium, and no other bacteria from outside can enter it. Before using the laminar airflow cabinet, we have to disinfect it either by using a disinfectant or using an UV light. And after that, before we begin the experiment, we have to switch on the air purifier, HEPA air purifier, for at least 15 to 30 minutes before, so that every particle inside the cabinet comes out. And when the cabinet is the air purifier is pumping out air, all the bacteria and dust particles cannot get in because it creates a positive air pressure inside. While working in a laminar airflow cabinet, always use sterile tools and petri dishes so that no contaminations enters the culture medium through them. Thank you.